Hey guys, in this video we're going to go over membrane transport, which is unit 1.4 of the current IB biology syllabus. Alright, so the cell membrane is a protective outer layer of the cell that allows transport. In other words, it acts as a barrier that controls entry and exit of all materials. It's selectively permeable, which means only certain materials can cross. Therefore, only certain materials are transported across the membrane. There are two types of transport: passive transport, which includes simple diffusion and facilitated diffusion, and active transport, which include endo and exocytosis and protein pumps. Let's start off with passive transport. Passive transport is the movement of substances across membranes with no use of energy. It's also called diffusion, which is a movement of particles from a region to another due to the random movement of particles. If you guys took chemistry or physics, you guys will know that all particles are constantly in motion, except for at absolute zero. Anyways, you guys don't need to know that for biology. More particles move down the concentration gradient, which means that more particles move from a higher concentration to a lower concentration. However, particles still move in both directions, just more particles move down the concentration. Anyways, net movement occurs until equilibrium is reached. And even when equilibrium is reached, movement still occurs. However, the amount of particles traveling from let's say place A to B and from place B to A are equal, and therefore the concentration of both sides don't change, for it is in equilibrium. Let's say that this line is the cell membrane. Obviously it would continue like that. This side is the inside of the cell, and this side is outside of the cell. So there are materials on the outside of the cell and on the inside of the cell. So, as you guys can see, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 particles outside of the cell and only 6 on the inside of the cell. Particles move in both directions. However, only 2 move from the inside to the outside, and I don't even know how many was that? 4 from the outside to the inside. Now, equilibrium is reached, and even though I didn't include an animation, particles would still be moving from both directions, just in equal amounts, so that the concentration of both sides stay relatively constant. Key takeaway here is that the net movement of the particles was down the concentration gradient, although particles moved in both directions. That's basically diffusion. Anyways, so as I said, there's two types of diffusion. The first type of diffusion is simple diffusion. It involves particles passing between the phospholipids, directly through the phospholipids in the membrane. The membrane must be permeable to the particles, meaning that the particles must be able to pass through the membrane. Examples include gas exchange in lung alveoli, which is highlighted on the left right there. So O2 moved from the alveoli to the capillary, while CO2, carbon dioxide, moved from the capillary to the alveoli. Another example is gas exchange in eye cornea cells. So oxygen goes into the cornea cells, while CO2 leaves the cornea cells. Oxygen gas, carbon dioxide, water, nitrogen gas, urea, and etc. are examples of particles that can pass between the phospholipids in the membrane. In other words, they're examples of particles that can be transported through simple diffusion. They're typically small in size because they need to be able to pass through the phospholipids. There are several factors regulating the rate of diffusion. 3 of them are proportional to the rate of diffusion, and 1, the molecule size, is inversely proportional to the rate of diffusion. So let's say if the temperature is high, the rate of diffusion will be high. If the concentration gradient is high, or in other words, if the concentration difference between 2 environments is high, the rate of diffusion will be high. If the pressure of the environment is high, the rate of diffusion will be high. However, if the molecule size is large, then the rate of diffusion will be lower than a smaller molecule. Osmosis is the simple diffusion of water. In other words, it is the net movement of water molecules from a region of low solid concentration to a region of high solid concentration. Just for review, the solute refers to the dissolved substance. So let's say salt water. The solute is salt, and water would be called the solvent. A hypotonic solution refers to a solution with a lower solute concentration. A hypertonic solution refers to a solution with a higher solute concentration. Now, avoiding osmosis in donor organs is crucial to organ transplants. So, let's say an organ is placed in a hypertonic solution. Since water moves from a region of low solute concentration to high solute concentration, organs placed in a hypertonic solution will lose too much water and therefore shrivel up. On the other hand, if the organ is placed in a hypotonic solution, it will suck up too much water and eventually burst. To prevent the bursting and swelling of organs due to osmosis, tissues and organs used in medical purposes must be bathed in isotonic solutions, which is a solution with equal concentration as human cytoplasms. Another type of passive transport is facilitated diffusion. Facilitated diffusion refers to the movement of substances across membranes through membrane proteins. There are a few examples of membrane proteins. One will be channel proteins. Channel proteins are transmembrane integral proteins that have very narrow holes. We went over this in Unit 1.3. If you guys want review, make sure you guys check that out. Anyways, channel proteins are selective. In other words, it allows only one type of particle to pass through. There are also carrier proteins. Carrier proteins are transmembrane proteins that transport molecules by changing its conformation. It's also selective, and facilitated diffusion is used by larger and polar molecules unable to cross through simple diffusion. And I forgot to mention earlier, molecules that are able to pass through simple diffusion are most likely nonpolar. The examples of particles that are transported through facilitated diffusion include ions, glucose, and amino acids. Potassium channels and axons is an example of channel proteins. So anyways, a nerve impulse involves the rapid entry of sodium ions and exit of potassium ions across the axon membrane. Potassium ions are charged, which means that it must go through facilitated diffusion and therefore utilizes potassium channels. The structure of potassium channels is as follows. It consists of 4 protein subunits, including the globular protein attached to the channel. It's selective to potassium ions, which means that it only transports potassium ions. Potassium channels are voltage gated, meaning that it opens and closes due to voltage. Now, negative voltage inside the cell closes the channel. This means that there aren't a lot of potassium ions inside the cell, for if there was a lot, the voltage would be positive because potassium ions are charged positive. Anyways, the positive voltage inside the cell. So once there are a lot of potassium ions inside the cell, the channel will open and the potassium ions will diffuse out, as like in this picture. Now, once the voltage reaches normal levels, the channel is blocked by a globular protein, like that, attached to the channel until the channel returns to its original state, which would look like this. Active transport. Active transport is the movement of substances across membranes using energy from ATP. If passive transport occurred down the concentration gradient, active transport occurs against the concentration gradient. Basically, it moves from a low concentration to a high concentration. Anyways, It allows cell to maintain different concentrations between intra and extracellular compartments. So the first example of active transport is endo and exocytosis. Endocytosis is the process by which macromolecules enter the cell by pinching off part of the membrane, causing the formation of vesicles. Phagocytosis is the intake of solid matter. An example would be the intake of bacterium by white blood cells. Penocytosis is the intake of liquids. An example would be kidney cells separating nutrients and fluids from urine. So basically that is a picture of phagocytosis. The membrane pinches in and consumes the solid particle. Likewise, the membrane pinches in penocytosis and consumes the liquid, fluids. Exocytosis is the process by which macromolecules exit the cells as vesicles move to the plasma membrane and fuse with it, releasing the contents inside. An example is insulin secreted into the bloodstream, neurotransmitters released at synapses. Exocytosis would basically be the same thing, except for this moves to the membrane and the fluid is released. An important thing to realize is that vesicles move materials within cells. The fluidity of membranes allows vesicles to pinch off or fuse with the membrane, and this allows vesicles to act as a messenger that transports materials. An example would be the transportation of protein. Make sure you guys know this. I'll just read over it, but make sure you guys internalize it and recognize the process, because most likely you guys will have to outline it. So vesicles with proteins produced by bound ribosomes bud off the rough endoplasmic reticulum and go to the Golgi apparatus. The Golgi apparatus modifies proteins, and from there, the vesicles bind off Golgi apparatus with the modified proteins and then go to the plasma membrane. The vesicles fuse with the plasma membrane, releasing the proteins by exocytosis. Another way to transport materials actively would be to utilize protein pumps, which are transmembrane carrier proteins. Protein pumps pump particles in a specific direction and transport only particular substances. An example of a protein pump would be the sodium-potassium pump. It looks like that. So the sodium and potassium pump maintains concentration gradients across the cell membrane. So this is how the sodium-potassium pump works. First, 3 sodium ions bind to a pump like this. Next, a phosphate group is added to the pump through the hydrolysis of ATP. It's okay if you guys don't know what hydrolysis is. Just think that ATP attaches to this protein and then it leaves one phosphate group behind. This causes a conformational change, or the change of shape, which releases the protein ions that were previously inside the sodium pump, or sodium-potassium pump. Then, from this conformation, 2 potassium ions bind to the pump. The phosphate group that was previously attached from this step is released. The release of the phosphate group causes a conformational change once more, returning the pump to its original state and releasing the potassium ions. That's it for today. Thanks for watching. Make sure to like this video and subscribe to my channel for more videos. Thank you.